Welcome friends, as a part of lecture series, today I am going to discuss the topic Microscopy and its types. Let us start with the history of microscopy. The early simple microscopes, which were only magnifying glasses, had one power, usually about 6x to 10x. It was Antony van Leeuwenhoek, a Dutch scientist and one of the pioneers of microscopy, who in the late 17th century became the first man to make and use a real microscope. Later on, Englishman Robert Hooke is credited with the microscopic milestone of discovering the basic unit of all life, the cell. Hooke is also credited with being the first to use the basic 3-lens configuration that is still used in microscopes today. The term microscope is generally used to refer to the type of compound microscope. In the compound microscope, the lens closer to the object to be viewed is referred as objective, while the lens closer to the eye is called the eyepiece. Now, coming to the basic components of compound microscope. The compound microscope uses lenses and light to enlarge the image and is also called an optical or light microscope. The compound microscope has two systems of lenses for greater magnification. First, the ocular or eyepiece lens that one looks into, and the second, the objective lens or the lens closest to the object. Now, let us know about microscope and its basic parts. First is eyepiece lens. The lens at the top that through which we look is an eyepiece. They are usually of 10x or 15x power. Second is tube. Tube connects the eyepiece to the objective lenses. Third is arm. Arm supports the tube and connects it to the base. Base is the bottom of the microscope used for support. Then is illuminator or light source, a steady light source used in place of a mirror. If microscope has a mirror, it is used to reflect light from an external light source up through the bottom of the stage. Sixth is stage, the flat platform where slides are placed. Stage clips hold the slides in place. If microscope has a mechanical stage, then it will be able to move the slide around by turning two knobs. One moves it left and right, and the other moves it up and down. Seventh, revolving nosepiece or turret. This is the part that holds two or more objective lenses and can be rotated to easily change power. Eighth is objective lenses. Usually, there are 3 or 4 objective lenses on a microscope. They almost always consist of 4x, 10x, 40x and 100x powers. When coupled with 10x eyepiece lens, we obtain total magnification of 40x, 100x, 400x and 1000x. To have good resolution at 1000x, it is needed a relatively sophisticated microscope with a condenser. Condenser lens. The purpose of condenser lens is to focus the light onto the specimen. Condenser lenses are most useful at highest resolutions, 400x and above. Then is diaphragm or iris. Many microscopes have a rotating disc under the stage. This diaphragm has different sized holes and is used to vary the intensity and size of the cone of light that is projected upward into the slide. Now, coming to microscope and its types. The invention of the microscope has brought about great inventions that have transformed the human race. This device is effective and very important in science because it provides opportunity for scientists to study natural elements that are not visible to the naked eye. There are different types of microscopes depending on the purpose for which it is intended. Microscopes can also be classified based on their image-making physical principles, area of application, and versatility. Now, let us discuss some major types of microscopes and their uses. First is light or compound microscopes. These types of microscopes are based on a simple principle of light and lens. A light source illuminates the object while the lens magnifies it so that it can be visible to the human eye for the purpose of studying or evaluating. Under this category are simple microscope, compound microscope, and the stereo microscope. Second major type of microscopes includes electron microscopes. These are the most advanced types of microscopes used in modern science. The electron microscopes are powered by a beam of electron that strikes any object that comes to its path to magnify it. Electron microscopes are used for studying cells and small particles of matter as well as large objects. Types of electron microscopes include transmission and reflection electron microscopes. Now coming to the microscopy. Microscopy is the technique used to view objects that cannot be seen by the naked eye but can be seen with the help of microscopes. There are three main microscopic techniques that are used, which include optical microscopy, scanning probe microscopy, and electron microscopy. First is optical microscopy or light microscopy. It involves the use of visible light and one or more lenses to produce an enlarged image of an object that is placed in the focal plane of the lens. This can either branch off into transmission, where the beam of light passes through the sample, or reflection, where the beam reflects off the sample surface, that is reflected light microscope. There are many applications of optical microscopy such as in nanophysics and biotechnology, but in medicine it is mostly known as being used in diagnosis when we are dealing with tissues or tests on free cells, which are known as smear tests. Types of optical microscopy. First is transmitted light microscopy. Transmitted light microscopy is the general term used for any type of microscopy where the light is transmitted from a source on the opposite side of the specimen to the objective lens. Usually, the light is passed through a condenser to focus it on the specimen to get maximum illumination. After the light passes through the specimen, it goes through the objective lens to magnify the image of the sample and then to the oculars, where the enlarged image is viewed. Now let us discuss some of the transmitted light microscopy. First is bright field microscopy. This is normal microscopy where no optical contrast technique is employed. It uses transmitted light to view a specimen that contains inherent contrast color or is stained. In order to get the best image possible from any transmitted light form of microscopy, and it is crucial that the light path be set up properly. The method for doing this is called illumination. It is also known as double diaphragm illumination because it employs both a field and an aperture iris diaphragm to set up the illumination. It simply works by allowing the light rays to pass directly through the eye without being deflected by an intervening opaque plate in the condenser. Second is dark field microscopy. In this type of microscopy, the specimen is illuminated obliquely with no direct light entering the objective. Features in the specimen plane which scattered light can clearly be seen against a dark background. Dark field illumination is provided by either a simple patch stop, a dark field element in a phase contrast condenser, or purpose-built dark field condenser. The latter is required for high-resolution objectives to prevent the oblique rays entering the wide aperture of the objective. Applications include detection of microorganisms in unstained smear preparations and classical diatom studies. Third is phase contrast. Phase contrast microscopy is devised by Frederick Zernike. This technique exploits the fact that light slows slightly when passing through biological specimens. The specimen is illuminated by a hollow cone of light coming through a phase annulus in the condenser. Phase contrast objectives must be used which have a corresponding phase plate. Light rays passing through the specimen are slightly retarded, and further retardation takes place in the phase plate. When these rays combine with rays which have not taken this path, degrees of constructive and destructive interference occur, which produce the characteristic light and dark features in the image. Fourth is polarized light microscopy. Polarized light microscopy uses plane polarized light to analyze structures that are birefringent. That is, structures that have two different refractive indices at right angle to one another. For example, cellulose microfibrils. Normal unpolarized light can be thought of as many sine waves, each oscillating at any one of an infinite number of orientations around the central axis. Plane polarized light produced by a polar only oscillates in one plane because the polar only transmits light in that plane. The polarized light microscope must be equipped with both a polarizer positioned in the light path somewhere before the specimen and an analyzer, and placed in the optical pathway after the objective rear aperture. Polarized light microscopy can be used to measure the amount of retardation that occurs in each direction and so give information about the molecular structure of the birefringent objects. Fifth is differential interference contrast microscopy. In this complex form of polarized light microscopy, two slightly separate plane polarized beams of light are used to create a 3D-like image with shades of grey. Wollaston prisms situated in the condenser and above the objective produce the effect, and additional elements add color to the image. Care must be taken to interpret differential interference contrast images, as the apparent hills and valleys in the specimen can be misleading. The height of a hill is a product of both the actual thickness of the feature and its refractive index. Variations of the DIC system are named after their originators, Nosepiece and de Sennermont. Options can be selected to maximize either resolution or contrast. These were all types of light microscopy. Now, fluorescence microscopy, which is second type of optical microscopy. Fluorescence can be used as a label or tag while preparing specific biological probes. Some biological substances such as chlorophyll and some oils and waxes have primary fluorescence, that is autofluorescence. However, most biological molecules do not fluoresce on their own, so they must be linked with fluorescent molecules, that is fluorochromes, in order to create specific fluorescent probes. The fluorochromes emit light of a given wavelength when excited by incident light of different wavelengths. Specimens labeled with a fluorochrome such as fluorescein or green fluorescent protein are illuminated with the relevant wavelength of light and emit the energy as a longer wavelength. The key feature of fluorescence microscopy is that it employs reflected rather than transmitted light, which means transmitted light techniques such as phase contrast and DIC can be combined with fluorescence microscopy. To view the fluorescence in the microscope, several light filtering components are needed. At the heart of the fluorescence microscope is the dichroic mirror cube, which comprises three components: a dichroic beam splitter, an excitation filter, and a barrier filter. Specific filters are used to isolate the excitation and emission wavelengths for each fluorochrome. Third is confocal microscopy. Although conventional light and fluorescence microscopy allow the examination of both living and fixed specimens, certain problems exist with these techniques. One of the main problems is out-of-focus blur degrading the image by obscuring important structures of interest, particularly in thick specimens. In conventional microscopy, not only is the plane of focus eliminated, but much of the specimen above and below point is also eliminated, resulting in out-of-focus blur from these areas. This out-of-focus light leads to a reduction in image contrast and the decrease in resolution. In the confocal microscope, all out-of-focus structures are suppressed at the image formation. So, these were all types of optical microscopy or light microscopy. Now, second main type of microscopy is scanning probe microscope. This is another branch of microscopy that involves using a probe to scan the object. Basically, it works by being moved around in a rectangular pattern known as raster scanning. A type of scanning probe microscopy is called STM, that is scanning tunneling microscopy. This is when a very sharp conducting tip is brought to the surface and a voltage is applied between them, and we are able to find out the tunnel current, and if this is maintained, we can trace the elevation of the surface and then produce it on an X-ray. There are advantages and disadvantages to SPM. However, for example, the major advantage being that small structures can be created as the interaction can be modified from the probe, and unlike electron microscopy, we do not need a vacuum. The disadvantages being that the maximum image is generally small and the detailed shape can be hard to distinguish of the scanning tip. Now, third major type of microscopy is electron microscopy. This is a form of microscopy that uses electron beams to create an image of the object being used. They have a much higher magnification than light microscopes and so a much higher resolution as a result. This allows us to see smaller specimens in greater detail. The resolution is able to be increased because as the electrons travel faster, their wavelengths become shorter. So there is a direct correlation between reducing wavelength and increasing resolution. Types of electron microscopy. All electron microscopes use electromagnetic or electrostatic lenses to control the path of electrons. The basic design of an electromagnetic lens is a solenoid, a coil of wire around the outside of a tube through which one can pass a current, thereby inducing an electromagnetic field. The electron beam passes through the center of such solenoids on its way down the column of the electron microscope towards the sample. Electrons are very sensitive to magnetic fields and can therefore be controlled by changing the current through the lenses. Now, transmission electron microscope. The original form of electron microscopy is transmission electron microscope, which involves a high voltage electron beam emitted by a cathode and formed by magnetic lenses. The electron beam has been partially transmitted through the very thin specimen, which carries information about the structures of the specimen. Partial variation is then magnified by a series of magnetic lenses until it is recorded by hitting a fluorescent screen, photographic plate or light sensitive sensor such as CCD camera. The image detected by the CCD may be displayed in the real time on a monitor or computer. Transmission electron microscopes produce two-dimensional black and white images. Second type is scanning electron microscope. Unlike the TEM, where the electrons in the primary beam are transmitted through the sample, the scanning electron microscope produces images by detecting secondary electrons which are emitted from the surface due to excitation by the primary electron beam. In the SEM, the electron beam is scanned across the surface of the sample in a raster pattern with detectors building up an image by mapping the detected signals with beam position. TEM resolution is about an order of magnitude better than the SEM resolution. Now, reflection electron microscopes. In terms of structure, the reflection electron microscope works much similar to the SEMs. Here, the reflected electrons are detected and gathered to study the surface of the specimen object. The REM are usually grouped with spin polarized and low energy electron microscopes to image the specimen structure. Last type of electron microscopy is scanning transmission electron microscope, which are also known as STEM. Just like the traditional TEMs, the scanning transmission electron microscopes pass an electron beam through a very thin slice of an object. The STEM focuses on the beam that passes beforehand and constructs an image through the raster scanning. Instead of focusing on the beam after passing through the object, it is a combination of high magnification of TEM and better surface detail of SEM. So, STEM is usually used to perform very complex analysis of objects and specimens that is not possible by just using the TEM. Thus, from today's lecture, we conclude that microscope is of crucial importance for observing microorganisms and understanding their structure. Microscopy provides valuable information about the functions of structures. Therefore, it is important to understand how microscope works. Further, there are various types of microscopes based on various principles such as light microscope, phase contrast microscope, confocal microscope, dark field microscope, scanning electron microscope, transmission electron microscope, and many more. Among various microscopic techniques, electron microscope has much higher magnification than light microscopes and much higher resolution. As a result, this allows us to see smaller specimens in greater detail. Thus, electron microscopy is considered as most efficient type of microscopy. These are the most advanced type of microscopes used in modern science. With this, we conclude our today's lecture. Hope you have enjoyed and understood it very well. Thank you.